Hello, and welcome to VetNeuroJR, the podcast where we unravel the mysteries of the canine brain, one fascinating case at a time. I'm your host, and today we're diving into a rather uncommon topic, cystic astrocytomas in dogs. These are brain tumors characterized by fluid-filled sacs within the tumor itself. While cysts in brain tumors are well documented in humans, particularly in a type of tumor called pilocytic astrocytoma, they are considered rare in our canine companions. To shed light on this subject, we'll be exploring a research paper titled Canine Cystic Astrocytomas, 7 Cases. This paper, published in the Journal of Veterinary Diagnostic Investigation, brings together the expertise of veterinary neuropathologists from the University of Georgia, Cornell University, and the University of Melbourne. They've compiled a series of cases that offer valuable insights into the occurrence, characteristics, and diagnosis of these unusual tumors. But where should we even start?
Let's begin by understanding what an astrocytoma is. Astrocytomas are tumors that arise from astrocytes, which are a type of glial cell in the brain. Glial cells provide support and protection for neurons, the primary signaling cells of the brain. Astrocytomas are a type of glioma, which generally refers to tumors originating from glial cells in the brain. So, what makes an astrocytoma cystic? Well, in simple terms, it means that the tumor contains one or more cysts, which are fluid-filled pockets. These cysts, sometimes referred to as macrocysts, are large enough to be seen with diagnostic imaging, like an MRI, or even during a gross examination of the brain. Now, here's where it gets interesting. In human medicine, macrocysts are a hallmark feature of pilocytic astrocytomas, a type of brain tumor that commonly affects children. In fact, cysts are found in over 80% of these pilocytic astrocytomas. However, in dogs, these macrocysts are not as frequently reported in gliomas, and are even more rare in meningiomas, which are tumors arising from the membranes surrounding the brain and spinal cord. This makes the study of canine cystic astrocytomas all the more important. Can you tell us more about the research?
The researchers behind this paper delved into the archives of veterinary diagnostic laboratories at the University of Georgia and Cornell University, searching for cases of canine astrocytomas with macrocysts. They also tapped into the extensive private archives of one of the authors, spanning over 4 decades. Their search yielded 7 cases of canine cystic astrocytomas. Let's take a closer look at these 7 cases. The average age of the affected dogs was 9.5 years. The study did not find any specific breed or sex predisposition. However, it's worth noting that 2 of the dogs were brachycephalic, meaning they had a short, broad head, like bulldogs or boxers. Sadly, due to the progressive nature of these clinical signs, euthanasia was elected in 6 of the 7 cases. The clinical outcome was not known for the remaining case. Grossly, meaning what was observed with the naked eye during necropsy, the tumors were described as tan to pale yellow masses that expanded and distorted the brain. In most cases, the tumors were well demarcated from the surrounding brain tissue, but in 2 cases, they were poorly defined. The location of the tumors varied, with 4 occurring in the cerebellum, 1 in the region of the thalamus and hypothalamus, 1 in the right putamen nucleus, corpus callosum, and lateral ventricle, and 1 in the left frontal and olfactory lobe. Importantly, all cases had distinctly visible intratumoral cysts, ranging in size from 2 to 10 millimeters, filled with clear fluid. In 1 case, secondary changes were noted, including cerebellar herniation, compression of the 4th ventricle, and obstructive hydrocephalus. When the researchers examined the tumors under a microscope, they found that most were low-grade astrocytomas. 1 case was classified as a high-grade astrocytoma. The tumors generally had low to moderate cellularity, meaning the cells weren't packed together very tightly. The neoplastic cells, which are the tumor cells, were arranged in bundles within a fibrillary stroma, which is a network of fibers. One interesting feature observed in some of the tumors was gemistocytic differentiation. Gemistocytes are astrocytes that have a characteristic appearance, with abundant, glassy cytoplasm and an eccentric nucleus. The cystic areas within the tumors were lined by neoplastic astrocytes and contained varying amounts of proteinaceous material, neoplastic astrocytes, foamy macrophages, and occasionally other leukocytes. The mitotic activity, which is a measure of how quickly the cells are dividing, was generally low across all cases. The high-grade astrocytoma did show some areas of necrosis and microvascular proliferation. To further characterize these tumors, the researchers used immunohistochemistry, a technique that uses antibodies to detect specific proteins in cells. They found that most of the neoplastic cells in 6 of the 7 cases showed cytoplasmic immunolabeling for glial fibrillary acidic protein, or GFAP. GFAP is a protein that is commonly found in astrocytes, and it's often used as a marker to identify these cells. It's important to note that intratumoral macrocysts are sometimes detected on MRI scans in dogs with glioma. However, these cysts are not always reported as a gross finding during autopsy. The findings of this paper emphasize that canine cystic astrocytomas are indeed rare. In fact, 6 of the cases were collected over several decades by one author, and only one case was found in the Athens Veterinary Diagnostic Laboratory autopsy archives over a 14-year period. Interestingly, in one case, the cysts were not mentioned in the original autopsy report, but were clearly visible when reviewing autopsy images. This suggests that these cysts may be underreported or overlooked during routine examinations. Now let's ponder the pathogenesis, or how these cysts actually form. The exact mechanisms are not fully understood, but several theories have been proposed. One possibility is that the cysts arise from vasogenic edema, which is fluid accumulation in the brain due to disruption of the blood-brain barrier. This disruption can lead to hypoxia, ischemia, and subsequent liquefaction of the tissue. Another theory suggests that cysts develop from long-standing edema and defective fluid resorption within the tumor. This can lead to entrapment of extravasated fluid and the formation of macrocysts from the merging of many smaller microcysts. It's also been proposed that microcysts containing proteinaceous material could coalesce and eventually become macrocysts. A final consideration is that macrocysts could be the result of chronic tumor necrosis, which is cell death within the tumor. However, in these cases, the high concentration of plasma proteins within the cysts suggests that blood-brain barrier disruption is a more likely mechanism than tumor necrosis. In human gliomas, tumoral cysts are typically lined by neoplastic cells. This is in contrast to cysts that may occur after surgery or chemotherapy, which are usually lined by normal brain tissue or dura mater. Unfortunately, there is limited information on the morphologic effects of chemotherapy and radiotherapy on canine gliomas. However, anecdotal evidence suggests that in dogs treated with chemotherapy or radiotherapy, the tumor site may be almost completely cystic and replaced by gelatinous tissue. Interestingly, a higher proportion of the cystic astrocytomas in this study were located in the cerebellum. This aligns with what is observed in humans, where pilocytic astrocytomas commonly occur in the cerebellum. The study also suggests that low-grade canine astrocytomas may be more prone to cystic change. However, further research is needed to confirm these associations and determine whether there is a link between cystic change, tumor location, and tumor grade. In conclusion, canine cystic astrocytomas are rare tumors with distinct features. They can occur in various locations in the brain, but may be more common in the cerebellum. The tumors are typically low-grade astrocytomas with a mixed neoplastic cell morphology. The presence of macrocysts can be a helpful diagnostic feature, but they may be overlooked during routine examinations. Further research is needed to better understand the pathogenesis of cyst formation and to determine whether there are specific factors that predispose dogs to developing these unusual tumors. That's all for today's episode of Vet NeuroJar. I hope you found this exploration of canine cystic astrocytomas informative and engaging. Thank you for joining me, and remember to keep those minds inspired, hearts light, and tails wagging.